Unit 3, Chapter 9, Biomolecules. In this chapter, we are going to see how to analyze chemical composition, primary and secondary metabolites, biomacromolecules, proteins, polysaccharides, nucleic acids, structures of protein, natures of bond, linking monomer in a polymer, dynamic state of body constituents, concept of metabolism, metabolic basis of living state, and the enzymes. First of all, 9.1, how to analyze chemical composition. We continue asking in the same way what type of organic compounds are found in living organisms. How does one go about finding the answer? To get answer, one has to perform chemical analysis. So we can take any living tissue, for example a vegetable or a piece of liver, and grind it into a trichloroacetic acid using a mortar and pestle. We obtain a thick slurry. If we were to strain this through a cheesy cloth or cotton, we would obtain two fractions. One is called filtrate, or more technically the acid soluble pool, and second retained it, or acid insoluble fraction. Scientists have found thousands of organic compounds in acid soluble pool. When we see about primary and secondary metabolites, most existing aspects of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure, and if possible synthesizing them. If one were to make a list of biomolecules, such as list would have thousands of organic compounds including amino acids, sugar, etc. for reason that are given in section 9.10, we can call these biomolecules as metabolites. Categories of compounds showing in figure 9.1, these are called primary metabolites. However, once one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, schemes, gums. These species are called secondary metabolites. 9.3, biomacromolecules. This is one feature common to all compounds found in acid soluble pool. They have molecular weight ranging from 18 around 800 daltons approximately. The acid insoluble fraction has only four types of organic compound, that is protein, nucleic acid, polysaccharides, and lipids. These classes of compounds are exceptions of lipid and having higher molecular weight in the range of 10,000 daltons and above. For this very reason, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those which have molecular weight less than 1000 dalton, and one those have higher, which are called macromolecules. Proteins are polypeptide. They are linear chains of amino acid linked by peptide bond, as shown in figure 9.2. Each protein is a polymer of amino acids. As there are 21 types of amino acids, a protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about amino acid content is important as later in your nutrition lessons. Next is polysaccharide. Acid insoluble pellet also has a polysaccharides. As another class of macromolecules, polysaccharides are long chain of sugars. They are thread containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a polymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animal have another variant called glycogen. Insulin is a polymer of fructose in polysaccharides chain. The right end is called reducing end and left end is called non-reducing end. It has branch as shown in the form of cartoon. Starch forms helical secondary structures. In fact, starch can hold I2 molecule in a helical portion. Starch I2 is blue in color. Cellulose does not contain complex helicus and hence cannot held I2. Next is 9.6, nucleic acid. The other types of macromolecules that one would find in acid insoluble fraction of any living tissue is nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecular fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is heterocyclic compound, the second is monosaccharide, and the third a phosphoric acid and phosphate. Next is structure of proteins. Proteins are mentioned earlier as heteropolymer containing string of amino acids. Structure of molecule means different things in different contexts in organic chemistry. The structure invariably refers to the molecular formula. Organic chemists always write a two-dimensional view of molecule while representing the structure of the molecule. 9.12, enzymes. Almost all enzymes are proteins. These are some nucleic acid that behave like enzymes. These are called ribozymes. One can depict an enzyme by line diagram. Any enzyme, like any protein, has primary structure, that is amino acid sequence of protein. An enzyme, like any protein, has the secondary and the tertiary structure. When you look at the tertiary structure, you will be noticing that the backbone of the protein chain folds upon itself and the chain rises across itself, and hence many sursives or pockets are made. One such pocket is active site. An active site of enzyme is sursive service or pocket into which the substrate files. Thus, enzymes through their active site catalyze reaction at a high rate. Enzyme catalysis differ from inorganic catalysts in many ways, but one major difference need mentions. Inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature. However, enzymes isolated from organisms who naturally living under extremely high temperature are stable and retain their catalytic power even at higher temperature. Thermal stability is thus an important quantity of such enzymes isolated from thermophilic organisms. Next is some interesting data about chemical reaction, and then the a short summary of nature of enzymes reaction is given below, and last is classification and nomenclature of enzymes. Let's see. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the types of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by the 4-digit number. Oxidoreductase dehydrogenase enzyme which catalyzes the oxidoreduction between two substrates. Transferase enzyme catalyzing a transfer of group other than hydrogen between a pair of substrates. Hydrolysis enzyme catalyzing hydrolysis of ester ether peptide glycosides. Lysase enzyme that catalyzes removal of group from substrate by mechanism other than hydrolysis leaving bonds. Isomerase includes all enzyme catalyzing interconvergence of optical, geometric, or positional isomers. Ligases enzymes catalyzing the linkage together of two compounds. Example enzymes which catalyzes of CO, CSCNPO, etc. bonds. We can see next topic is about cofactor. If you want to study an enzyme and cofactor in detail, we have a separate video lecture. You can see above the I button is seen. You can click there and see exact detailed lecture about the cofactor and enzymes related. Thank you.